Hello, everyone. My name is Haytham Alsayed. I'm going to be presenting my research for the 2025 Spring Honors Symposium titled Nucleocytoplasmic Shuttling of Thyroid Hormone Receptor Alpha-1 Acetylation and Non-Acetylation Mimics. So some background for this research. Thyroid hormone is a very ubiquitous hormone in the body. It affects a lot of functions, primarily metabolism, and it can affect energy levels, also developmentally, and in children, it's got important functions for growth and development as well as neurological function. Um, and the way this works is it binds to a receptor called thyroid hormone receptor, which travels into the nucleus and binds DNA to either activate or repress target genes to affect a response. So it's basically turning genes on and off for a particular function. And thyroid hormone receptor, or TR, in this case, TR alpha 1, it's just a subtype of TR, undergoes a process called nucleocytoplasmic shuttling, which is just transport in and out of the nucleus. And we can see in the graphic that it shows this mechanism here. And it uses these things called importins and exportins, which are just proteins that help it get through the pores and nucleus. So TR alpha 1 is reversibly modified by acetylation, which is the addition of a chemical group onto a certain portion of the protein. And this occurs in a nuclear localization signal, which is the part of the protein that the importin binds to help it get into the nucleus. And we've seen in previous research that in the acetylated form, TR alpha 1 has a greater presence in the cytosol than the non-acetylated form does. But it's not clear whether nuclear transport of TR alpha 1 is affected by this acetylation. So what I'm researching and the question that I aim to answer here is, does TR alpha 1 acetylation impact its ability to undergo this shuttling mechanism? So the method we use to do this is a little bit complicated, so I'm going to try to explain it as simply as possible here. Basically, what we're doing is we're creating a hybrid cell that has a nucleus from both a human and from a mouse. And the way we're doing this is we're transfecting, or we're introducing a little piece of DNA that has the protein that we want to express. So either the natural version, the wild type version of TR alpha 1, or what we call mimics that are mimicking the chemical properties of the acetylation and non-acetylation mimic. So we add that little piece of DNA to a human cell, which is labeled HeLa here, and we let that sit overnight so it can express the proteins that can accumulate in the cell. And then we treat it with something called cyclohexamide to stop any new protein formation. So any proteins that are hanging out in the cytoplasm at this point should be transported into the nucleus. And then we add our mouse cells to the same slide, and we fuse them using polyethylene glycol, which weakens the membrane so it makes them come together and fuse like that. You can see this big cell here that has the two nuclei in it. We let that sit for between 1 to 12 hours. Usually 1 hour is enough to allow the protein to shuttle completely and reach equilibrium. About one hour is about generally how long that takes. But it can be left to incubate for as long as 12 hours if we need that for timing purposes. Then the cells are killed and they're stained with two separate stains, sorry. And we image them with fluorescence microscopy to see if we have any shuttling. You can see in this very complicated graphic on the right, which I'm going to explain in a second. We have green showing up in both nuclei in all three rows. So the top row is the wild type, which is the natural form. The middle row is the acetylation mimic, and the bottom row is the non-acetylation mimic. And we can see that we have green in both nuclei, so we can take that to confirm shuttling. The blue stain here is just helping us distinguish between the human and the mouse nuclei, and the red stain is showing us the shape of the cell, so we can tell that the two nuclei are in the same cell, and not just two separate cells that are really close together. So future research we can do to investigate these localization patterns of acetylation and non-acetylation mimics, we can either we could characterize the kinetics of import and export for these two forms, basically characterize how long it takes for them to get in and out of the nucleus. And if those rates are different, they can affect where it accumulates. We can identify factors related to nuclear retention. So is it binding to structures in the nucleus and staying there and not being able to be exported? And then identify factors that affect the lifespan of both forms. So what I mean by that is how quickly are they degraded? How long are they present in the cell from being synthesized to being degraded? Where are they degraded? Are they degraded in the nucleus or the cytoplasm, etc.? So these are just some future studies we can do to further explore this phenomenon. And that's a brief overview of my research. I hope you enjoyed, and thank you for your time.